All right, today we're going to do Lab 17, the blood typing lab. Unfortunately, I'm not here today, so I want to go through the information that you would get if I was present. So the first thing on Lab 17, if you can make one minor adjustment in the pre-lab, the third line, it says A negative. If you can change that to A positive, that would be great. To then answer the pre-lab questions with your partner to go through a review of what blood typing is and how you achieve a certain blood type. So the procedure, there is two groups, a group A and a group B, because the number of bottles, all the bottles that you need for today are located over underneath the clock side of the room. You should see them already. They're lined up. Any bottle that is behind the bottle in front would be the same type. So for example, there are a group of bottles that have last names on them. Here's one of the examples that you would have. This is Mr. Smith. You need to know what's inside this bottle. This bottle contains purified red blood cells. So not his antibodies. We've kind of purified them so that only inside of this, it's clear solution, obviously a simulation of blood type, but what's in here are the red blood cells. And from here, you're going to be able to determine what letter blood he has, whether he has A blood, B blood, AB, or O. So what you need to do and grab as well, so you'll have four names, or four bottles that have names on them, but you'll also need two bottles, one that says anti-A, and one that says anti-B. The anti in these bottle names represent antibodies. So just like how the names have purified red blood cells, what's inside this bottle is purified antibodies. So you have to look in your notes if you want to recall like how we simulated what an antibody looks like. So that's what's in this bottle. So what you'll do, I'm going to grab a bottle that you never have to work with. This is another name that is not in your lab, but Reed, for example. What you'll do, you'll take one microscope slide, which is found on the window side of the classroom in the labeled box that says microscope slides. I'm going to put a drop on the left, drop on the right. So then, notice how this is clear, and all the solutions that I've actually been showing you have been clear or colorless. So what I'll do is I'm going to drop a drop from the anti-A onto the left-hand side. I'm not going to dip the tip of the medicine dropper into the solution already there. That would cross-contaminate. So from a distance, I'll put a drop on. Nothing changed. Notice it kind of still says, you can still see the white paper underneath. So then I take the B, the anti-B, and put some antibodies that are anti-B onto the right side. So from the view, you probably can't see much. So I'm actually going to change the background to give you a better sense that the right side here, keep it on the right, so this one did change color. I had two colors, now I have a different color on the right. This one stayed the same. So that's important. That's what you're going to be putting into your data table, is recognizing when and where the color change occurred. So from this information, that simulated change right here indicates a clot occurred. So you have to refer to your notes as to what would cause a clot to occur when someone that has A blood gives to someone with B blood. That's not what we did here. We didn't give someone's blood type to another person. So it's a different way of thinking you have to kind of understand, using the same knowledge base of how blood-to-blood transfusions occur. Well, how did this lab simulate that? The red blood cells from these bottles were mixed with antibodies from these bottles. Sometimes they clot, sometimes they don't. So you have to find out, what does that mean? What type of blood would that Reed bottle have, which is the bottle I put down first, based on that outcome? Next, we're going to then decide, well, what letter or what positive or negative type blood do they have? If you refer to your lab, you will see that there is a list, that there's a number bottle associated with each of these names. So for example, this bottle 1 is associated with Mr. Smith. I use the letters of the last name to identify if they have A, B, AB, or O. I'm going to use the numbers to identify if they have positive or negative blood. To help me do that, there are plastic bottles over there. They're not all labeled, but it's the only one that's in plastic form. This is anti-positive, which means it's antibodies that are they fight the positive antigen. So that's what you would need for this. There are four numbers over there, so you're going to grab 1, 2, 3, and 4, and then you're going to grab one of these bottles. You shouldn't have both sets of bottles in front of you. There's not enough to go around for every single person. So what I'll do, if I then assumed that in my example, you don't have to do number 5, but I'm going to show you what you would do next. I'd clean up my microscope slide and I would put a drop, and I just need one today for this section. I don't need two. Then I will mix a drop of the antipositive onto this single drop, and nothing happened. All right? So this was a no change scenario, so it didn't change white. There is another color that it possibly can change, and you'll see that throughout the lab. So again, I have to interpret that. What does that mean for the blood type? Does he have positive or negative blood based on that information? So you're going to do that. This is a tricky lab. It's more time consuming to analyze the data as it is to actually just put drops of the different letters onto the different slides. So to recap, you only need 4 slides to start. If you grab the names first, then you figure out the letters of their blood type first. If there's not enough dropper bottles to go around for all the pairs, and you have to work with the numbers first, that's perfectly fine. You can figure out if a person has positive blood before you figure out if they have A blood. That doesn't matter. You'll swap eventually, and then you'll get the second set of bottles, and you'll get both pieces of what you need. All right? So the real trick here, and I'm sure some of you will be asking others in the class as you do this lab, you really have to understand what causes clots to occur. What does that mean then for the blood types of the individuals on these slides? So I hope that helped, and you have the rest of the period to work together to figure out the outcome of this lab. Good luck.